Okay, welcome back, guys. So today we are going to talk about what is blood group, and this will be the our introductory portion. And this video lecture series will be divided into 18 different parts, where we are going to talk about ABO blood group in very deep and details, right? So before going to the main topic, we need to talk about some basics of antigen antibody reaction, right? We all know what is antigen and antibody is. Antibody is a immune component that is produced by B cell that play an important role in human immunity, right? And antigen is any foreign particle. Antigen may be ourself. We are also going to talk about this, right? And between this antigen antibody there can be a formation of reaction, right? And that means antigen and antibody have the ability to react with each other, and there can be two type of antigen antibody reaction depending on the type of antigen. If antigen is particulate in nature, then reaction of antigen and antibody leads to agglutination. If antigen is soluble in nature, then reaction of antigen antibody leads to precipitation, right? So our classification of ABO blood group is typically focused on agglutination reaction, right? So who first discovered this ABO blood group? ABO blood group was first discovered by scientist Karl Landsteiner. He discovered the ABO blood group at the beginning of 20th century by examining the agglutination reaction between RBC of some individual and serum of another individual. He detailedly note the pattern of agglutination between RBCs of some individual and serum of some other individual, and found out that our blood group can be divided into 4 different groups, that is 1A, 1AB, 1B, 1O, and another one is AB, and that is our main focus part. And this type of classification of blood group is known as ABO blood group system. Another blood group system is MN blood group, that is not our part of discussion. So before going to talk about this, let's focus on this picture. This picture going to tell you everything about ABO blood group system that you need to know, right? So this is a typical vein where blood is flowing. This is RBC, and this is blood plasma. Remember another one thing, that our blood can be divided into 4 different parts, that is RBC, WBC, platelets, and blood plasma. Among them, blood plasma will conquer 50 to 55% of total blood, and RBC will take 40 to 41%. Approximately 1% will be our WBC, and less than 1% will be platelet, right? So this is this is our blood plasma, and this is our RBC. In the surface of RBC, our antigen is present. Now आप लोगों को ये जानने की इच्छा होगी कि एंटीजन इनसाइड द बॉडी क्या कर रहा है, और इसमें हमारा एंटीबॉडी भी क्यों नहीं रिएक्ट कर रहा है, राइट? Before going to talk about this, we are going to want to talk about where antibody is located. Antibody is found in the blood plasma. The antigen is found on the surface of RBC, and antibody is found on the blood plasma, right? And now let's talk about this. Why this antibody do not react against this antigen. Before going to talk about this, you need to know that antigen can be of two types. One is self antigen, that is produced by our own body to train our immune component to fight against non-self antigen, right? And another one is non-self antigen, that is coming either externally or can be a result of or can be a result of cancerous infection or malignant infection, but that is not a part of antibody, because in that time cytotoxic T lymphocyte will take charge. So those guys are non-self antigen, An antibody will typically react against this, right? It will not take any charge against self antigen, and that is what we know as immune tolerance. एंटीबॉडी कभी भी सेल्फ एंटीजन के अगेंस्ट कोई भी एक्शन नहीं लेगा, क्योंकि सेल्फ एंटीजन इज आवर बॉडीज ओन, right? Self antigen take part in T cell development, where it play a important role train or educate the T cell to fight against non self antigen whenever it is required in the secondary lymphoid organ, right? So that is why self antigen is important, and it is made by our own body, and antibody do not take any charge against this self antigen. If somehow the self antigen become attacked by antibody, then we will call that particular condition as autoimmune disease. So that is the idea, that is the reason why the antigen present on the surface of RBC is not attacked by antibody present on blood plasma. You only need to know about if we are talking about blood group, you only need to that antigen is present on the surface of RBC and antibody is present on the blood plasma. That is the idea. And in that case, in terms of biochemistry, antigen can also be referred to as agglutinogen, and antibody can be referred to as agglutinin, right? We can call antibody as agglutinin. ओके, सो हम लोगों ने क्या जाना कि ब्लड इज ऑफ 4 टाइप्स. वन इज आरबीसी, one is WBC, one is platelet, and another one is blood plasma. Blood plasma acquire 50 to 55% of the total blood. RBC will acquire 40 to 44%. Platelet will be less than 1%, and WBC is near about 1%. In the surface of RBC, there are antigen present, and in blood plasma, antibody is present, right? And that is the idea of overall blood, and that play a pivotal role in blood grouping, right? So as I have previously told you that blood group can be divided into 4 types, that is A blood group, B blood group, O blood group, AB blood group. The blood group will be considered as A when it contains antigen A on the surface of RBC, right? That means in the surface of RBC it will have A if the blood group is A, and in that case the antibody will be its opposite, that is anti B, right? Similarly, in case of B, the antigen will be what? B, and the antibody will be anti A. That means the blood groups are typically named on the basis of the what type of antigen present on the surface of RBC, right? Now I know you think that in case of O the antigen will be O, but that is not the case. In that case, no antigen will be present on the surface of RBC. We will also talk about that there are some H antigen in later videos when we are talking about the development of all this type of antigen, how this antigen develop. We will talk about in details about that in later videos, right? So in all the blood group there is no antigen on the surface of RBC, but anti-A and anti-B antibody will be present on the plasma. And in case of AB blood group, there will be antigen A and antigen B on the surface of RBC, and no antibody will be present on the blood plasma. So that is all about the introductory portion of blood group, where we have learned that blood group can be divided into 4 different types, that is platelet, WBC, RBC, blood plasma. And blood group they can be divided into 4 types on the basis of type of antigen present on the surface of RBC, that is blood group A, blood group B, blood group AB, and blood group O. Blood group AB or blood group system was discovered by Karl Landsteiner at the beginning of 20th century by examining the agglutination pattern of some individuals RBC and some individuals serum, right? And it is a type of agglutination reaction. So that is all about blood group, and it is our introductory portion. We will talk about in much more details about ABO blood group in our upcoming videos. So I think if you find it helpful, and if you, then please hit the like button, share with this video with your friends, and subscribe my channel. Thank you.